Good morning. My name is Jamie Haddock. I am a computational and applied mathematics assistant adjunct professor at UCLA. It is my privilege to introduce Professor Bonnie Berger, who is the Simons Professor of Mathematics at MIT, holds joint appointments in electrical engineering and computer science, and is associate member of the Broad Institute. Professor Berger received her AB in computer science, magna cum laude, from Brandeis University, and received both her SM and PhD in computer science at MIT, where her thesis won the George M. Sprouse Prize for Best Research Contribution to Computer Science. She is a pioneering researcher in computational molecular biology and was instrumental in defining the field. Her work includes seminal contributions in diverse fields, including compressive genomics, network inference, structural bioinformatics, metagenomics, and genomic privacy. Professor Berger's numerous awards are far too many to list, but a few include election to the American Academy of Arts and Sciences, the NIH Margaret Pittman Director's Award, the Biophysical Society's Dayhoff Award, selection to Technology Review Magazine's inaugural TR100, an NSF Career Award, an honorary doctorate from EPFL, and fellow of ACM, ISCB, American Institute for Medical and Biological Engineering, and American Mathematical Society. Without further ado, it is my pleasure to welcome Professor Berger.
So, good morning. Much of my work has focused on ideas that center around transforming and creating algorithmic thinking to new areas of computational biology. As I will talk about today, we will use or leverage modern algorithmic techniques to design computational platforms for biomedical data sharing and analysis at scale. And hopefully, some of the problems I talk about will inspire you because we're really counting on the mathematics community to help us out. So back in 1995, when I first started working in this area, we thought these were big numbers, but they're not anymore. And around that time, I went looking for biological problems to work on on the suggestion of my postdoc advisor, Danny Kleitman. And I ran into a biologist, Peter Kim, and he posed to me the following problem. Can you predict coiled coils, that motif on the screen, from sequence, from just the sequence? And after many meetings with me learning biology and Peter algorithms, we were able to predict coiled coils by use of pairwise residue correlations. And I have to say that I was quickly able to give him a proof of concept that these ideas would work, and I think that that really helped because I kept his interest. Anyway, so now everything has changed. As you can see on the screen here, genomic data is growing exponentially faster than both computing power in blue and disk storage in red, and green, I mean. Genomic data is in red, computing power is in blue, and disk storage is in green. And back in 2002, where this plot starts, we didn't even have such a plot because it wasn't a problem. But it's become increasingly a bigger and bigger problem, and that's what I'm going to talk about today. In fact, biology used to be biology, but now biology is inseparable from algorithms. And in fact, to do leading edge biology, you really have to leverage modern day algorithms. So in addition to its massive structure, massive scale, this biological data is siloed all around the world in independent repositories. These are dispersed through academic labs, biobanks, and companies. So today, I'm going to focus on sequencing data, that's DNA sequences, and RNA transcriptomic data in the form of single-cell data. By pulling together these rich troves of biomedical data, we can empower scientific advances. unprecedented ones, in fact, as you're going to see. But the problem is that with this massive data comes many computational challenges. Sorry, I'm having a little trouble here. So our vision is to give researchers access to this massive dispersed genomic data. And I'm going to get into detail as to how we do that. And this is all toward the democratization of data so that someone on their own laptop has access to the same data that someone in a massive institution like the Broad has. So the idea we follow here is we want to compress the data in such a way that we take advantage of the low-dimensional structure of biological data manifolds. And fortunately, these do exist, and I will show you those as well. So first, I want to start with an area we introduced, in fact, with my grad students, Michael Boehm and Po Rouleau, while they were math grad students at MIT. They've both moved on. And it's called compressive genomics. So given that the growth in sequencing data that I just described, one has to wonder, how are we going to process and store this data in algorithms that we would like to use to analyze it? And clearly, if you just wanted to store the data, then you would compress it. And for that, you can use programs like gzip. Okay. And traditional methods for compressing data have only been somewhat successful in a biological setting. There's so much data that even the Beijing Genomics Institute sends their data around on hard drives. So biologists, in fact, had stopped at that point. They thought, okay, if we want to store and transmit our data, we will compress it. But we raised the next question in 2010 or 11, and that was, you have all this data, but eventually you have to look at it to analyze it. So in this little schematic here, you compress the data, but you have to uncompress it to analyze it, and that takes a lot of time and storage. So we don't really want to go that route. In fact, that route won't even work. So we came up with this notion of what we called compressive genomics. And here, it allows you to compress the data and operate directly on the compressed data without loss of accuracy. So this comes in handy, because what if you want to search across your data, or you want to run statistics on your data? Then what we want to do is structure our compression so that we can do search and compression simultaneously. And for that, we structure our biological data so that we can compress it in such a way that we can search it easily and quickly. So the inspiration for this is we realize that much of the data we've seen is similar to what we're going to see. And this is, in fact, because of evolution. We realized back then, 10 years ago, that there was redundancy in our data, and that should be helpful in compressing it. So here you see a subtree of the fly phylogeny, also known as Drosophila. And we get the size of the individual genomes colored independently by the relative length of these bars. And the size of the non-redundant data is colored in black. Okay, so if you look up here, my mouse won't work, but if you look up here in this upper corner, D-simulans and D-Seychellia are two extremely identical genomes, so the amount of unique data between them is equal to just the size of one of the genomes. And as you get more and more data, the savings in compression only grow. And you do get more and more data in this sense. So that's the basis of the idea. So let's view redundancy in our data in a different way. Suppose we take all our genomic sequences and we view them in some vector space. Now, this is going to be a very high-dimensional space. And the interesting thing about it is that we realize this data doesn't occupy the entire space. In fact, it occupies a very small portion of this space, which can be shown in this tree-like structure here. Now, this is just a 2D visualization. It's not a two-dimensional projection of our data. We'll see those later in the talk. And so the nice thing here is that we can cover our data with, I hope you can see them, a relatively small number of circles or spheres. And this, we can use those to represent our data. And notice that one of these spheres is going to have a lot more points. If one of these spheres has a lot more points, then we'll have to use fewer of these spheres to cover the data, or if many of these spheres have a lot more points. Okay, so I'm back over here. So this is probably known to you as the metric or Kolmogorov entropy of the data. That is the number of covering spheres it takes to cover our data. But now we're going to get to the newer realizations. And I'm going to exemplify this by something called search of this data. And that is akin to sorting in computer science. Biologists use search all the time. Computational biologists use search all the time. And the reason is they want to know if a sequence is similar to one in their database, because that gives them a lot of information. Okay, so that green triangle there is our query. And we want to search within a given radius of our query. We want to know all the points, all the genomic sequences, or long subsets of, contiguous subsets of them, which one of those are close to our query or within this given threshold. So let's say that's our given threshold. And let's say we want to do a cluster-based similarity search. We'll first do a search over a representative from every single cluster. And this will be a coarse search in the sense that we'll have this outer circle, which is where we actually search out to. However, the problem is if there are a lot of clusters in that outer circle, then the algorithm's going to take a long time and it won't work. So, or a lot of these spheres. But we're very fortunate that our data has what we call low fractal dimension. That means when we look out or we scale out to neighboring spheres, there are not too many of them. And in fact, fractal dimension is usually 1 or 2. So in particular, let's just look simply at this problem and why the algorithm works. Okay, so we have a bipartite graph of queries to sequences in a database, and both are genomic sequences here. So we want to find all the matches within a given search radius to sequences in the database. And every single point is a sequence in the database. And of course, the query is a point. So believe it or not, biologists used to look for through every one of those blue points before we did our work to find sequences of interest. But if we took inspiration from that previous slide, and we were able to structure our data in such a way that we did not need to search all our data and without loss of accuracy. Okay, so these Voronoi diagram components are essentially our covering spheres from the previous slide. And we clustered our database originally on the right side of the bipartite graph into similarity clusters, and the red ones are the cluster representatives. So then, all we need to do is search through the cluster representatives to find matches of interest. And in fact, we do, as I said, a coarse search on those within a slightly relaxed radius, which the outer circle represents. And I apologize to people on the left side of the room, but my mouse clicker doesn't work, so I'm over here. So anyway, so we do a coarse search on a looser radius to find clusters of interest, and then we're gonna brute force the search only in those clusters of interest, which are shaded in blue. Okay, so we're given the search radius, the inner circle, but we want to know how we choose the outer circle for the coarse search radius. So we're going to look at a little blow up of this region here, and we realize that if our data, or points between our data, obey a distance metric, then we can use the triangle inequality. So very simply, we have our query point, and we know that it's within the threshold distance radius from a point that matches it, and we know that that point is within the radius of a cluster of some cluster center. So we're going to use that information. So we have that radius, and then, of course, by the triangle inequality, we will make the more relaxed search threshold equal to the threshold radius distance plus the radius of the cluster distance. Okay, now given that, we also were able to prove that the triangle inequality is not required. And this becomes important because biologists use something called E-value, which doesn't quite obey the triangle inequality, but is close enough. And in fact, what we were able to prove is that if our points usually obey the triangle inequality, then in high likelihood, we will find all the matching points. Okay, so what is the runtime of this algorithm? Well, we have to pre-process our data. If there are endpoints in our data set, that's order n, but since genomes don't change too much, we don't have to do that too often. And as new genomes come along, they're so similar to the ones that we've seen that we can amortize their addition to our entropy scaling structure. So let's focus on the actual runtime of the algorithm, not the pre-processing one. And what we're going to do is combine both our notions of metric entropy and fractal dimension. So we know that our coarse search is proportional to the number of covering spheres, which is the metric entropy of our data set. So we only need to search metric entropy number of those cluster centers that I showed you. And then our fine search scales with a fractal dimension of our data set. And of course, all these will be dependent on the data set. They'll be different for different data sets. And then of course, we have to output our results. Now, as I said, it turns out the fractal dimension is usually between 1 and 2. So the metric entropy term here dominates the runtime, and we can say that our runtime is linear in the entropy of the data set, and that's the metric entropy. We can also show a similar result for space, but there we need to use the information or theoretic or Shannon entropy of the data set. And I won't have time to go into that here because I want to get on to all this exciting new data that we're getting, a different kind of data. So what were we able to do with this? Given that I'm an applied mathematician, I like to see what we can do on real data. And in fact, our coil-coil programs are now being used or have been used by thousands of people. They have like thousands of citations. So you can really make an impact in this field, even with low-hanging fruit, if you do something. So there's this program, BLAST, which everyone uses for search. It's the most popular program in the field. And when we looked at 36 yeast genomes at the time, that was a small number, but that's what we had in about 2011. And we saw that BLAST's runtime, typical ordinary BLAST, scaled linear with the amount of data, and that our coarse search scaled almost sublinearly, and our full compressive search scaled close to that. At the time, we didn't have enough data, so we simulated more genomes and found that both our compressive and coarse search scaled sublinearly, as we expected, with the size of the data. But now we have a lot more data, and we know they definitely scale sublinearly with the size of the data. So we also did this for BLAT, another souped-up version of BLAST. And don't worry about this data anomaly here. It's just part of the data. It's present in both algorithms. And what we were able to find is we got the same results. And in fact, similar to with BLAST, we were able to find at least 99% of the matches that both BLAST and BLAT found with our compressively accelerated versions of them, which we called KBLAST and KBLAT. So we have algorithms that scale sublinearly with high sensitivity on real genomic data. And in fact, this is the last slide on this part of the talk, we were able to extend this to protein sequences at the same time in the initial work. And then after that, in subsequent work, we were able to extend this to all sorts of biological data because we could cover it with a small number of spheres, metric entropy, and all this biological data had low fractal dimension of 1 or 2. So there weren't too many neighboring spheres we had to search. And in fact, all the different things we could do is we could compressively speed up protein structure search, metagenomic sequencing search, that's, for instance, from the gut, the microbiome, and other areas. We were also able to search reads and compress their structure, and also small molecules from PubChem. And we got extremely similar results for all of these. And in fact, the cool thing is the programs we accelerated, the original programs that thousands and thousands and thousands of people used, we could actually use those straight out of the box for the coarse search. And then we could do our brute force search of the surrounding area, and we got nearly the same findings as these other programs, which took a lot more time. Okay, so we've seen how sublinear time algorithms can be really effective in analyzing data we can compress. And now we're going to turn to a really new, exciting area of data that's technically been around for 10 years, but has really only taken off in the last 4 years. And in fact, we jumped into this area like 2 years ago, and we're already known as like experts in the field. So this is how fast things are moving in this field. So you can't sit there forever as a mathematician and puzzle over the most perfect proof here. You've just got to throw something out there, and then later you can do your proofs. Okay, so this exciting new area moves from primarily a sequence component to a more geometric component. And what we want to do is we want to sketch this data in similar to the way that we compressed our other data. So I'm going to talk a little bit first about what this data is. Okay, so what made this data possible is that sequencing a genome is becoming really cheap. And in the last 10 years, the cost of sequencing a genome and the speed has far outpaced Moore's law, to the point where the original Human Genome Project, embarrassingly, was given $100 million. And I was involved in some of the first efforts to sequence the first human genome as part of this consortium. And now we're down to only $1,000 to sequence a 3 billion base pair genome such as yours. Okay, so with this lower cost, enabled us to ask biological questions that we couldn't even dream of before. I mean, originally I used to think like, how do cells differentiate in the body from embryogenesis to adulthood? Or how do cancer cells metastasize? And what are the different phases of that? But we couldn't interrogate our data to get answers to that. But now we can. And we do it through this single cell technology. And the way this works is for a tissue, you have lots of cells, and here I have similar ones colored the same. And what we used to be able to do is only analyze the average gene expression from all the cells. So we could see for each gene over all the cells its average expression. But now what we can do with this new and exciting data is we're given all this single cell data as readouts, and actually it's done with RNA transcriptome sequencing. And what we can do now is that we can measure the gene expression for every single cell separately. And that allows us to do all sorts of really cool things, including what biologists like to do is project this data down to the first two principal components, or first two what they call t-SNE components, and they love to visualize their data, and they call that the gene expression space. Now, projection is nice, but I'm not a huge fan of it because it actually does not allow you to look at your data on these higher dimensional or low dimensional manifolds. And as I said, much of our data lives close to a low dimensional manifold, as you saw in that cartoon there. So with this new technology has come a slew of studies, and the different technologies are colored separately. And this is only growing and growing and growing, as exponential as you can get. And now we are up to profiling 2.5 million cells over thousands and 10,000s of studies from all around the world. And there are consortia, such as the Human Cell Atlas, which is probably the most famous one, that seek to profile 10 billion cells over the next years. Okay, and all of this is in the last 10 years. So I'm gonna talk to you about two problems in this area, and then I'm gonna move on to some open problems that hopefully you can help with, because we're counting on the math community to help. And there are a lot more problems to solve. And this is very interesting data, so I think for geometers, it really provides a place where you can apply what you know. And any help is welcome in biology and computational biology. Okay, so the first problem I'm going to focus on is integration. That is combining our data across multiple experiments. So as I've represented here are like 3 different experiments, 3 different studies. They can be across tissue types. They can be across cell types. They're from different institutions and different studies. Okay, so what we want to know is what are the common cell types among these studies, and are there interesting patterns that will tell us something more about biology? Okay, so previous to our work, there were algorithms for integrating data, but they could only integrate 2 of these studies if they knew a priori that there was a cell type in common between the studies. And that's all they could do. And later I'll show you some plots on how they do and why they didn't do so well doing that. So our approach was to use a technique that your camera uses for panoramic scanning. So we applied panoramic algorithms for integrating multi-experiment heterogeneous data. The second problem I'm going to talk about is, given this massive amount of data and integration, you get more and more data, comes more scalability issues, like I was speaking about earlier. And current algorithms are only designed for very small scale uses. So we had to come up with new algorithms. And in fact, the single cell analysis has to look at thousands of genes measured in hundreds of thousands and millions of cells. Okay, so that's a lot of data. So single cell analysis is inherently geometric because we're dealing with many points with very high dimension. So again, we took inspiration from compressive genomics, and we tapped into a fairly new area of computer science called sketching, which Piotr Indyk, right near me at MIT, does. And so our approach was to select an informative subset of cells called a sketch, so that we could operate on only that sketch without loss of accuracy. And this should sound familiar. Okay, so let's return to the integration problem. So this is kind of what the data looks like. Look at the top left. We're looking at two totally different technologies. And if the data is uncorrected, when we project it along the top two principal components, we only separate by technology, which is not informative at all. And even when we labeled our projection by cell type, we found that similar cells were not clustered together. And we definitely want common cells to be together. So what we were able to do with our algorithm is run on many different cell types, like brain and blood and et cetera, different studies that provided those. And we were able to, quote unquote, correct this data so that it wasn't separated by technology or confounding factors, and it actually clustered similar cell types together. Greens together, orange together, purple together. Okay. And so in that way, we were able to compare across many diverse and heterogeneous data sets to do this. This is not just one study, as you see two there, but we could do many. Okay, and as I said, the way we did this is through panoramic stitching. So the way that's typically done is you have all these separate images, and you look for commonalities of the images, and you put them together to get a final picture. And that's a picture of this data center, which I am partly housed in and at MIT. And that would be your stitched panorama. So what we aimed to do was find a way to stitch together this single cell data by finding commonalities of cell types across all the different studies at the same time. And the way we did this was, which allowed us to make progress where others had not, and now the field is starting to adopt our vision. So what we did is that we combined mutual nearest neighbors only across all the subsets. And then what we did was we accelerated our algorithms by doing things like PCA, as I said, I wasn't so happy with, and also locality-sensitive hashing. But that allowed us to do amazing things with real data. And that was just our first attempt, but there is so much interesting mathematics here that it's just unlimited what you can do, especially nowadays. Okay, so one result we got was we did single cell, and notice that we called our algorithm Scanorama for single cell panorama. So it's all these kind of cute names in this field. So we looked at 26 completely diverse data sets, diverse across tissues and technologies, and these we were able to combine with, and these are blood, I think they're blood cells. They're blood cells. Actually, they're all sorts of cells. They're different tissues, technologies, they're everything. Sorry, I'm getting to blood cells later. And as you can see, our program Scanorama on the left here was able to cluster similar cell types together. And the data was spread apart by these commonalities and patterns between cell types, which you can see here. However, the other program, which I clicked on too soon, in the middle column here, those were two programs that were really well used at the time, SCRAN-MNN and Seurat-CCA, and we found that they overcorrected the data. They squashed it all together, and they didn't cluster similar subtypes of cells or similar cells together. So Scanorama was really effective at that. And now I'm going to turn to the scalability problem. Because while Scanorama was good, it wasn't as fast as we needed it to be to handle millions and even possibly billions of cells. So just like with compressive genomics, what we want to do is we don't want to look through every single cell in our database, as I explained. We want to be able to cluster these cells so that we can look into sketches, so we can just operate on the cluster representatives in this case. And we don't even do the brute force surrounding search. And we get nearly the same results. So this is more lossy, and the other was lossless compression. So we're going to leverage, again, the structure of our data to effectively sketch it. And fortunately for us, again, this new kind of data also has low metric entropy, which is sublinear in the size of the data for this too, and low fractal dimension, in fact, less than 2. So one way we can sketch this data, a very simplistic way, is we could just do uniform downstream downsampling. Okay, so if we uniformly downsample the data, then the problem is we get these red points are the sample, the blue dots are the cells, and we get way too many of the common subtype. And as it turns out, not enough of the more interesting ones that span a greater and more diverse biological space. And the reason these are more interesting is they're much harder to obtain. They can be rare subtypes, which uniform downsampling misses. And what those are is those are the small number, for instance, of stem cells in a regular tissue, or they're the cells that have migrated from their tissue of origin and are metastasizing in cancer to other cells. And we can now track that with this new kind of data. But uniform downsampling misses that, and we can't use it. Okay, and would you believe, in 2018 in Cell, biologists were still manually, and I know this because they would hire undergrads to do this, they were labeling all the same cells and similar cells in their experiments so that you can read right there that there was a better balance in their data so that they could find these rare and different, not so common cell types, which are the ones that hold all the interesting biology. So one approach we tried 2 so far, is related to the case center problem. So here we wanted to downsample according to the geometry of the data. So one thing we noticed is, as I said before, when I showed you my tree of life, suppose I call it, was that if cell types are very similar together, then they're going to have low metric entropy because we don't need so many spheres to cover them. However, the ones we're interested in have high metric entropy because they're distributed along a bigger biological space. So what we decided to do is these red ones would be the ones supposedly in our sketch, is we formalize this by tapping into the Hausdorff dimension, which you're probably familiar with. But what that says is what's the worst case distance between an element in your data set and one in the sketch. So in this case, our elements are cells. So we want to minimize that. And so we want to find a sketch with the lowest Hausdorff dimension, but unfortunately, that's NP hard. So we had to turn to approximation algorithms. And here's one of the ones we considered. It was a greedy algorithm, just like greedy algorithms do really well on some classes of NP hard problems, like set cover and vertex cover. We decided to try a greedy algorithm here. And the way it works is you first randomly pick a point to be in the sketch, and that's labeled in red as a red dot. Then you pick the point that has the greatest Hausdorff distance from the sketch, in this case, the first point. And you should be able to see it in red here. What went wrong? Anyway, there it is. That's the farthest point on the upper right. And then we keep doing this. And you see another point up there in the middle, and then, so here we finish up, and we get a sketch of k equals 6. And we have ways, intelligent ways of determining k, but I'm not going to have time to go into that here. But when we're all done with this, we can prove that we get a sketch which is within twice of optimal, but it can be somewhat slow because we have to look through all these points to measure the Hausdorff distance at each iteration of our algorithm. So then we tried something which I like a little better, which we called geometric sketching, and we called our algorithm GeoSketch. And so what we really want to do with sketching is not just detect the rare cell types, but also detect those that have complex transcriptional structure, as it's called. And those are in green here. They're not kind of like all in a ball, they're covering the space. They may not be so rare, but they have that interesting structure. And we don't want to try to find the common cell types. And the reason that this is so difficult is because this data has batch effects and other confounding factors, and we have to overcome them. Okay, so we developed this notion called a Plaid covering, and essentially what it does is it covers the interesting cell types with uniform gray boxes or hypercubes, because remember, this is multi-dimensional data, and you can see them shaded in gray here. And our data is covered by all these uniform boxes. And so the way we do this is we sweep from left to right here across the Plaid covering, across our data actually, one dimension at a time. And when we discover a point, we put a uniform gray square around it, or hypercube, and then we just keep going. And as you'll see here, when we don't have points, you'll have more space between the columns, let's say, or the rows. Okay, so that's what a Plaid covering is. And then compared to geometric sketching, compared to uniform sampling, you can see that uniform sampling misses a lot of the points we want from the rare or uncommon regions, and geometric sketching uniformly captures those. And the way we sketched our data was we picked one representative from each of those gray boxes that we used to cover it. So we're able to achieve the same result as Hopper, the greedy algorithm, in linear time, or nearly the same result. And we can do this if the fractal dimension is low and constant. And it turns out that for single-cell data, it is. So we could also use our algorithms to visualize the data. And so here we have a lot of brain cells. And as you can see, uniform sampling just doesn't find the rare ones. And as an example, geometric sketching clusters similar cell types together better and finds rare ones, such as macrophages and microglia. We're also able to do better data integration, not just visualization. And GeoSketch is able to dramatically accelerate our Scanorama program. Here's the original data. Here's what Scanorama would do with it, and it looks pretty good. And that took 2 hours. But if we GeoSketch it first and then run Scanorama, we even get what kind of looks like a better result in only 8 minutes. We also have other methods for data visualization of single-cell data. So now I'm going to get to the really cool thing here and then tell you a couple open problems. The cool thing is that we can get insights into innate immunity. So what I mean by that is that there are currently these things called macrophages, and they're very important in immunity. And they have an anti-inflammatory state shown in blue and a pink one shown in, or a pink one that stands for an inflammatory state. So currently, we have macrophage protein markers, and they can't distinguish state in a binary way. They kind of go from high to low, and we thought we should be able to do this. We have protein markers for T cells, also immune cells, where we can distinguish state. So what we did was we ran Scanorama on all of this data, and then we performed differential expression on the Scanorama clusters to find unique marker genes, ones that no one really had identified before for distinguishing state. And here on the left is just a pictorial diagram of what it would mean to not distinguish state on the top and distinguish state on the bottom. State A is a state we're interested in. It's the inflammatory state. So what we did, well, the biologists were working with Brian Bryson, and my student, Brian He, shockingly enough, can do experiments. But you're getting a new generation of like mathematicians who can also do experiments. But they checked these protein markers in actin, which is a very common protein, and they checked against this new gene we found, ALOX5AP, which is well known to be involved in fact, necessary for lipid signaling. However, no one had identified it as a marker gene for macrophages. And we were able to see that we could distinguish. This is how biologists do these plots. We can distinguish between orangish and aqua, only for ALOX5AP. We also hit it with a pathogen, and only the inflammatory states popped up. So we've discovered some new biology, which is very, very good. It feels good. Anyway, so the point is, Scanorama aids in revealing critical elements of lipid signaling and macrophages. Okay, open problems very quickly since I have to finish up. Noise models for manifolds, we need them. We know that our points live near a low dimensional manifold, and it has lots of points on it, but we get these new observations, and where do we put these points? Well, if we had a noise model, potentially we could distinguish the outliers that belong in that noise model and those points that just need a new manifold. that should be on a new manifold. And finishing up, biologists are also interested in these trajectories of developmental data. Here you're looking at blood cells. We have 3 types of blood cells across, we have blood cells, lots of blood cells, across 3 tissues that are given in the upper right. And we basically, they all start, they all start as progenitor cells here in that triangle there in the lower left, and they branch out into 3 separate arrows, black arrows, as they distinguish into myeloid, lymphoid, and MEP types of tissues. Okay, so one thing we want to do is figure that out. We have ideas how to do this. In fact, it relies very much on things like sketching. We actually look at the co-expression matrix for each of the clusters that we get as a result of sketching, and that's on bioRxiv and under review. And the point is, it allows us, and before people had not looked at co-expression across multiple groups of cells or clusters. They had only done differential expression in single cells or co-expression in single cells. So this is a different model, and it allows us to effectively represent diffusion pseudotime, or these trajectories that biologists love. And also, they are the most informative trajectory. So the last thing I'm going to show you is another result where we looked at neuronal development. We have a timeline from these studies from early mouse development to adult development. And we were able to absolutely, using our algorithm, our co-expression algorithm, which we call CoScape, reproduce the developmental trajectory from mouse neuronal embryogenesis to adulthood. So one question I have as an open question is, can we come up with a parsimony-based model that is the simplest explanation is the real one, like Occam's razor, and that's used a lot in phylogeny analysis. And in fact, Ingrid Dobesky's talked about this yesterday. So can we come up with a parsimony model of how a manifold would, quote unquote, split in 2? So I want to thank all those students of mine at some point who worked on compressive genomics, started by Michael Bayman-Peru, and the entropy scaling search was done by William Yu, who's now at Toronto, Noah Daniels at URI, Rhode Island, and Dennis Yurikoglu at Google Search, and both Michael and Peru are now at Harvard Medical School. And then I also want to thank for all the single cell work, everybody who's in my lab, Brian He, Ben DeMeo for the sketching, Hoon Cho, an amazing guy who just became a Schmidt Fellow at the Broad, which is really prestigious. Sarah Nyquist, who's done a lot of the work in single cell and can also do experiments. And also Rohit Singh, who's returned as a research scientist to my lab, and Brian Bryson, who's in bioengineering at MIT and is an experimentalist. And I want to thank my entire group. And I'll show you the group one more time. They're more important than the software. Anyway, everybody supports everybody, and we have a very happy group. Anyway, and here's the software that we've developed. So I'll now take questions if there's time.
Thank you, Professor Berger, for that wonderful and inspiring talk. I believe we're actually out of time, unfortunately.
No problem.
So we'll take questions offline, Professor Berger.
I'll be around.
Great. Thank you.